Thank you everybody for joining us today. We have another great pitch event for you. We have 5 great companies and a few of our great panelists. Just want to remind everybody in the audience, if you do have a question, go ahead and put it in the chat box. I just unlocked it so you can type freely in there. We'll do our best to get to the questions, but like I said, we do have a tight schedule, so we might not get to them and answer them offline, but we'll try our best. And with that, I'm going to hand it off to Hall, our CEO. Hall, go ahead.
Great. Thanks, Shane. And I want to thank everybody for joining us today. I want to thank our panelists that are going to be speaking today. If we could, let's have our panelists introduce themselves first, and then we'll go into the companies that are pitching. We have 2 investors here. First up is Eric Alford. Eric, can you tell us more about yourself and your fund?
Yeah, sure. So I run a fund called Kmont Ventures, and we focus on seed through Series A investing. In addition, I'm also a board member and vice president for the Houston Angel Network. We're one of the largest and most active angel networks in Texas.
Great, thanks. And with us also is Laura Hamilton from Spring Mountain Capital. Laura, can you tell us more about yourself and your fund?
Yeah, I work at Spring Mountain Capital, which is an early stage growth equity fund based in New York. So we invest across the A and the B rounds, leading and co-leading within digital healthcare and B2B technology.
Great. Thank you guys for joining us today. We also have investors in the audience, and those in the audience, we welcome you to put your questions into the chat box, and we'll do our best to answer them today. If not, we'll follow up afterwards to make connections as well. We will poll everybody for interest at each deal, so be looking for that as one of the best ways to connect with the startups that we have. So with that, let's go and kick into our first presentation. Each one gets 15 minutes to pitch and answer questions. So we'll have a timer up here. So first up is SynJam. We have Jackie Iverson. Jackie, thank you for joining us. And if you'd like to go ahead and share your screen, thank you for joining us, Jim. We'll go ahead and have you guys kick off first.
Great. My name is Jackie Iverson. I'm the founder and chief clinical officer for SynJam Pharmaceutical. I'm joined with my CEO, Jim Iverson, and I will be presenting on SynJam, and afterwards you can ask either of us questions. SynJam Pharmaceutical is a company that got started to reduce inflammation. We have a platform of agents to do that. And we do this by incorporating mast cell modulators into products for both the over-the-counter and prescription strength. Our platform is based on the problem that more than 50% of all deaths worldwide are attributed to chronic inflammation, where 61% of Americans have chronic inflammation. And over the course of the last 3 years, or last 4 years, Americans have lost 3 years of life expectancy. We cannot continue at this rate. And what's prevalent amongst a huge number of people is that they are suffering with the interconnected consequences of inflammation. Inflammation can exist in both the acute, the chronic phase, and causes tissue damage that is irreversible. This leads to aging, premature aging, tissue damage, disease, and death, and disability. And what we might not realize with this interconnected part is that there are a number of different things that affect us that have to do with runaway inflammation. And what we're trying to solve for is this runaway inflammation that we have found is involved with COVID-19 or upper respiratory infections, with alcohol hangover. Believe it or not, that is an inflammatory response, not dehydration. And more specifically, or also, is opioid use disorder. When a person withdraws from opioids, an immediate immune response occurs. And we have a platform of agents to address all of these other indications that we are going after, but our 3 lead assets are for upper respiratory infection, alcohol hangover prevention, and opioid use disorder. This is the biggest advance that can happen with investment, can happen in this entire space. Right now, we have consumers who are looking for something to reduce inflammation, but the only things on the market are either products to reduce pain and some inflammation, but they don't do it through the immune system of reducing inflammation through the very beginning of the inflammatory cascade, where the mast cell resides. We are using products that incorporate a mast cell modulator into them in order to prevent the mast cell from degranulating and causing an inflammatory response. And these products can reduce the inflammation that occurs at other end organs. This allows people to have something to reduce inflammatory pain or just plain inflammation without immunosuppression, where other products cause immunosuppression. This is a huge advance for the consumer. And again, we do this by modulating that mast cell to prevent it from releasing the 4 main cytokines of inflammation, which affects the rest of the body. The placement of our products would be right after supplements, right after behavior modifications, for example, changing your diet, changing exercise, increasing your health, or decreasing inflammation. But you can only get so much out of supplements, out of food modification, out of your environment change. And some people are genetically predisposed to inflammatory or chronic inflammatory disease. Our products would be used right after you tried everything else in the over-the-counter market and would fit either over-the-counter for acute inflammation, or you could use our products in the prescription strength for more potent anti-inflammatory release or potential. We have 8 papers published, 4 university collaborations, 12 therapeutic assets, and over 40 patents. We have a corporate support strategic partnership with KVK Tech, who is manufacturing our 2 products at this point, one for our upper respiratory infection and one for our prevention of alcohol hangover. The market is huge because inflammation exists in so many different disease states and different pathologies that are affecting most people. And the only things on the market, as I said before, are those things that cause immunosuppression or don't do a good enough job of reducing the inflammation from the immune system. This is our pipeline. I want to emphasize the 3 lead assets, again, for COVID-19, because it decreases inflammation and protects lung damage. We can use it not only for COVID-19, but also other upper respiratory infections. You could get this on board right away and then test yourself to see if you have a viral infection, COVID-19, influenza, or the common cold, and you could continue to take our product to reduce inflammation and protect the lungs, and you could add an antiviral to that. Our alcohol hangover is a 1 pill you take prior to drinking alcohol, and with biomarkers, we can show that we can reduce that inflammatory response. And also opioid use disorder. We have garnered the interest of NIDA, and we're meeting with them next week to see how they can help us with our development plans. So getting these 3 drugs to the market, and these are just 3 of our platform, you'll see a huge interest by the consumer in wanting to take our products because nothing exists in these spaces. Our competitive advantage is that we are particularly well tolerated. These drugs are extremely safe. They don't cause immunosuppression, and they prevent systemic inflammation. And we are on a mission to make sure that these drugs are affordable so that we can democratize healthcare. The consumer does not have a product available to decrease inflammation by the mechanism of action that we have. We have a great team of entrepreneurs and innovators who have already shown their worth with greater than $700 million in exits. We have raised $7.5 million in funding to date, and we are now looking to raise $10 million to advance those 3 clinical assets to the end of phase 2 and seek licensing deals. Thank you.
Thank you, Jackie. Let's go to our investors. Eric, What is your first question?
Hey, Jack, interesting concept. I'm curious if you could talk a little bit about any rebound concerns you have when patients stop using it. Because I can see how, decrease inflammation, but, once a patient stops, is there the concern that it comes back even stronger or higher or, more aggressive?
Yeah, so our first products are for acute inflammation, and with those products, we hope to get them on the market, and those can help us solve for exactly what you're curious about, as we are curious too. There is, as we start manipulating our immune system, and this is new science and new research, we have these questions too. And at the same time, we know that these agents can also decrease inflammation in a lot of other organs, the heart, the metabolic system, the brain, the kidneys. And so we too want to explore that. And to do that, we first need to get these products on the market into patients, using them for acute inflammation to see how they're going to affect them if they started taking them for longer than 2 weeks.
Okay, thanks.
Laura, what questions did you have?
Yeah, so I'm more curious about the funding side of the clinical trials, given that you're only raising a 10 million Series A. So I guess how to fund the rest of the trials, and kind of the realistic estimate to get the drugs through FDA approval. I know that one of our drugs in our portfolio, I think, was 200 million. It's pretty high.
Yeah, well, this is one of the innovations to our platform, but I'm going to have Jim answer that question.
Yeah, thank you, Laura. Great question. So we, first of all, we're combining small molecules that already exist. So we're making a new chemical entity, not a new molecular entity. So when I hear numbers like 200 million, I start thinking of new molecular entities, and how we've been able to do this is through a lot of innovation. So we're currently in a phase 2 clinical trial with Duke University, where we're doing the trial in Kathmandu, Nepal. That trial much lower order of magnitude, but in the United States we could have done it for about 5 million. We're doing it in Kathmandu, Nepal for $1 million, and that'll get us to the end of phase 2. And so one of the things that we're looking at doing right now is we have these 3 lead assets that are all at or in phase 2. So the oral COVID therapeutic is in a phase 2 study. The product for reducing inflammation around alcohol consumption, we're doing a clinical trial in Australia, and we could do that trial for about $2 million. That'll be a phase 2 trial, and then one of the great things about Australia and doing that trial in Australia, the Australian government has a 43.5% cash R&D rebate. So the first $1 million that we put up, they will give us $435,000 back, and we have a partner that's willing to loan us against that right out of the gate. So we're only looking for, on the first, like I described in the first tranche here, it's actually only $5 million of the $10 million Series A. We broke it down into 2 tranches, of which we got a $2 million commitment from our corporate venture partner, and then so we're actually looking for 3 million, and that 3 million will complete the oral COVID therapeutic, complete into phase 2, complete the alcohol hangover phase 2, and then also we're doing a study around opioid withdrawal to open up a phase 2 study, which will cost a fair amount of money. But as Jackie had mentioned earlier, the National Institute of Drug Abuse has recently reached out to us in an effort. They've seen our early research, and they would like to get behind us and see how they can support us to bring that product through its clinical and regulatory development.
Great, thanks. My question, Jim, is this is a platform-based solution. What are the other applications you envision for it down the road?
I'll handle that question. So this technology is really needed, and I can provide research articles upon research articles of how the majority of disease states that we have today we're finding out have an inflammatory component to them. And the way we're doing research and science to address those chronic inflammatory diseases is one organ at a time in a silo. And we're attacking like one biomarker at a time. Whereas our mechanism of action is to decrease the amount of inflammatory mediators that come out of the mast cell, which is at the very beginning of the inflammatory cascade. So our product could be used across the board for helping and being an add-on therapy to current disease states. So there's a great potential to use this across the board for cardiovascular, for atherosclerosis, for diabetes, for neuroinflammation, et cetera. And so it's huge potential in the market. And although we can't grab all of them all at once, we're trying to do them pinpointed in certain indications so we can show the benefit of the products.
Great, thank you. I believe we're at the end of our time here. If anybody in the audience has a question, please drop it in. If not, we'll go ahead and close up this one and talk to the next one. With that, let's go ahead and looks like we're not getting a question yet. So let's go ahead and move over to the Creative Biotherapeutics, Donald and Anita Davidson. If you could go ahead and let's go ahead and get your slides up and move to the next one.
Thank you for the opportunity to speak. I really appreciate it. My name is Dr. Don Davidson. I'm the CEO and co-founder of Creative Biotherapeutics. We're a biotech company that is delivering novel therapies to treat drug-resistant diseases. Our motto is we succeed where other therapies fail. So we are raising Series Funds A round funding to complete our drug scale-up and our rat toxicity study to move to the FDA. 3 million will complete that, and then we'll follow on with the second close to do 5 million to do our phase 1 clinical studies. So what do we do? Well, I created and patented a first-in-class anti-cancer and anti-COVID therapy known as CBT-300. And I know what you're saying, both therapies in one? Yes. So CBT-300 stops resistance of recurrent cancers and cures severe COVID infection. Currently, there are no effective therapies for these patients. The problems we want to solve are recurrent drug-resistant cancers and severe complex COVID infection. So is there a need? These are the top 10 cancers in the US last year, and you can see breast, lung, and prostate are always in the top 3. And the white bar shows the number of those patients that have recurred. Now, these patients have failed all other therapies. It's usually like 4 or 5 different therapies, and they have failed all these therapies. That's where our drug will work. And if you look at the circles, which I've drawn here, which show the effectiveness of our drug in these different cancers, that's over a $21 billion market annually for our drug. And our goal is to enter the market in metastatic breast cancer in the second quarter of 26. For COVID, does it still exist? Yes, it actually does. You don't hear much about it, but this chart is from the CDC themselves. And the black line is the number of deaths per week. And I've just looked at the data today, and there's still around 1,200 to 1,400 patients dying a week from COVID, not with COVID, but from COVID, and that's around 200 a day. So there's still a huge market and a number of people dying in the US from COVID. If you just take those patients, that's over a $700 million market annually. We also know that our drug is effective against long COVID to reduce the effects and to reduce the side effects. And so that's a market we didn't even add here at this time. So what's our solution? Our solution is CBT-300. And I designed it to be an anti-cancer drug. And at the same time we were doing that, COVID hit. And we noticed that viruses use the same mechanism to evade the immune system that cancers do. Well, our drug affects that evasion and allows it to see both of these diseases. And so we're developing CBT-300 for both diseases. So how's it work? Yes, it will work for both. So this is my diagram. I can show you a bunch of data and everything, but this is the diagram of how it works. So we all know that tumor cells are different from normal cells and they change. But what most people don't know is that when we give them chemotherapy, when we give them targeted therapies, those cells change again and they change for much worse. And what they do is they put this protein on the surface, a stress protein called GRF78. Now that causes a cascade of events to make them drug resistant, immune resistant, and nothing is going to affect these cells when that finally reoccurs. What our therapy does is simple. It removes that stress protein, GRP78, from the cell surface. And when that happens, the chemotherapy and the immune cells can come in and attack, eliminate the tumors, and you're left with a normal cell again. That's great for cancer. What about virus? Well, when you have a comorbidity like lung cancer or asthma, really bad asthma, or anything that affects that, what you do is normal cells will put GRP70 on the surface. It's a stress cell. That's what the mechanism is to do, is put that on the surface. And we and others have found, and we actually got a National Science Foundation grant, only 2 companies out of 20,000 to get the grants, was that the GRP70 acts as a doorway for the viruses to get inside the cell. If you block that doorway, the viruses can't get in. I don't care how many times they mutate, we're affecting the cell and not the virus. And so now the immune cells don't do anything. If we take away that GRP78, the cells revert back to a normal phenotype, the viruses are cleared from it, and you have a healthy person. If there's no GRP78 there, 80% of the viruses can't get inside the cell, of all viruses, not just COVID, of all viruses. So as a platform therapy, CBT300 solves both severe COVID and recurrent drug-resistant cancer by normalizing the cells. Does it work? Here are 4 very aggressive drug-resistant cancers: melanoma, lung, renal, and breast cancer. We ran these as a blinded study in mice with an immune system, and we ran it against Keytruda, the mouse version of Keytruda. And as you can see in each one of these, we're superior to Keytruda as a standalone therapy. Does it work against viruses too? On the left side, you'll see that we ran against COVID, influenza, and Ebola, a lethal dose of each of these viruses, different companies, all blinded, showing that our therapy caused 44% COVID cures, 50% influenza and Ebola. And we ran the COVID at a very late stage. We waited 3 days before we started dosing. So yes, it's very effective and works. How about competition? Well, for metastatic breast cancer, our therapy checks off all the boxes from low cost to it's a biologic, so it's a single injection once a month. Drug resistant, cold and hot tumors means there's their immune system or not, and it's non-toxic. Now, the great thing about that is the GRP78, which is the target of our drug, is 99% homologous to the human version. Therefore, we're really, if we see no toxicity in mice, which we haven't, we're not too worried about toxicity in humans. And let's be clear, I know you're never supposed to do this. You always put competition down, but there are no approved therapies for late stage severe COVID, nothing. So I can't put it down. We'd be the only one in the class right now. So our advantage, well, CBT utilizes decades of expertise in my drug development and my co-founder's commercialization, resulting in less risk, less cost, and less time. What do I mean? Well, it's been published that it takes 10 people, 10 years, and $100 million to find a target, validate the target, find a drug, find the lead optimization, do everything. We've done the same thing with 3 people, 4 years, and $2 million. So less cost, less risk, and everything. My name is Dr. Don Davidson. I have a PhD in biochemistry. I have 33 years experience in Abbott and AbbVie for drug development and moving 4 drugs from the bench to the clinic. My co-founder is Anita Davidson with a doctorate in physical therapy. She's run a clinic for the last 25 years. She's founded 4 other companies that have been very successful. She is truly an expert in commercialization and project management. We run a decentralized model where we don't want to bring all this overhead in. So we're doing CROs to do a lot of the heavy lifting. And many of these CROs we've already vetted from when I was at Abbott and AbbVie, and she's vetted when she was at her clinic. And we have drug manufacturing, distribution, commercialization, and Syneos is our partner for doing regulatory and our clinical studies. We do know that as the drug moves along, we're going to have to bring more and more of that talent in-house. We'll have to pay for that. And so we'll extend our internal staffing for marketing, sales, and finance as it goes along. Our intellectual property is strong. We had a 5 composition of matter patent for 5 compounds in 2021. We have 2 use patents that are moving through review and have gotten good marks from the office actions. Our pipeline is we have our CBT300 that works very well for viruses. We expect to go into the market first with COVID-19 and to also go with breast cancer. Now, the phase 1 clinical, we can do disease patients. So what we're going to do is cancer patients that have infections. So we'll use the same population for that phase 1. And then in phase 2, we'll decide to go either for COVID first, which will be much faster than cancer, and go from there. Our return on investment is 48 times. Return on investment is our estimate, and that's looking at the different EBITDAs and return on investment. As you see, you add them up for the 5 years. We get that from our price per dose of 5,000. Our cost per dose is $650. Our CRO says they can reduce that 2 to 3-fold, but that's the price for manufacturer, purification, bottling, sterilization, shipping, and the whole thing, and to put the label on it. We expect a dose of 1 to 2 per patient for COVID, and probably about 12 doses per year for cancer. As you can see, we expect the $8 million to do our drug tox and our drug scale up, to do phase 1 studies, and then we'll come back with the second round to do our phase 2 studies. We have prior funding. We got a $1.3 million in NSF grant. We've applied for 2 other grants of breast cancer and COVID, $3.5 million each. We have expect to do a raise of $3 million to do our 3-month rat tox and our drug scale up, and then do a second close to do a $5 million for the phase 1 clinical study. Exit possibilities are the equity reverts back to CBT when the ROI has been returned. We could continue with equity dividends long-term, or possible licensing the drug or several drugs in 2029 to 30, or potential IPO or buyout. So we have strong intellectual property through 2041. Our investor in value is 48 times in 5 years. We have 1 therapy for multiple diseases. Our product is safe and non-toxic, easy to deliver, works in several different diseases. Our competition, we're superior than so far at this point, and there's no approved for late stage COVID. We don't chase the disease, we protect the cells from the disease. Thank you for considering for support, and here's our addresses here on the left. Thank you.
Thank you, Donald. Thank you, Anita. Let's have our first question come from Laura on this one. What would you like to ask, Laura?
Yeah, so typical investor question. I'm curious, just given the earliness of the drug and still focused on the animal studies, what's the rationale for giving dividends starting in mid 2025, like around 2 years from now?
The dividends for given the 2025 is that's when we believe we'll be in the market, and we are, because the market's so huge, we're taking out of that what we need to do continual clinical studies, what we need to do drug scale up, what we need to do other stuff, and that's our dividends from that, and that we are going to return that to our investors early so they can get return on investment. We're not taking a salary. We're going to take it from the dividends.
Great, thanks.
So if I can, Laura, good question. Can I just tag on to that? When looking at the pro formas that really, we've been extremely aggressive with making sure that we have enough money retained back with the EBITDA, and with the combined markets, we feel that we can really offer those types of dividends and still be very successful and very profitable. But we feel very strongly about our investors, and we feel if we make money, they should make money.
Great, thanks. Eric, what questions did you have?
Yeah, actually, I'm curious if this, I guess I was trying to understand. So with this, so say if a cancer patient took this, does that also then, if they came down with COVID, would that protect them from COVID? So is it kind of a one use for any type of virus, or how does that, or is it customized per virus?
No, I think the virus dose may be a little bit higher than the cancer dose, but yes, if you're taking a dose that can affect the virus, it should affect both. Absolutely, 100%. We actually like to do our phase 1 with cancer patients that have COVID, so we can see if we do see a decrease in virus and if we get any hint of a decrease in tumor load.
Okay, thank you.
Great. And my question is around the reimbursement rates with COVID solutions like this. What kind of reimbursement are we seeing right now?
That's a great question. The reimbursement rates we're seeing are much higher than what we have. Remdesivir is much higher for reimbursement, and they're doing it. The antibodies that used to be effective are no longer effective. We're higher or about the same as this. And so the reimbursement rates, we have an expert that's working with Blue Cross Blue Shield, and he says that we're right in the ballpark. We could even go higher with our costs.
Great. And Eric, Laura, did you have another question to ask?
I was just curious, just to follow up on the kind of the question that Laura asked on On the exit, would you be able to peel off, say, the cancer treatment or the COVID treatment early for an even quicker exit for investors?
Yes, so that's a yes with a caveat. So forgive me for having the caveat, but I worked at Abbott and AbbVie for 25, 30 years, and I know we used to buy drugs just to shelve them. And so the caveat is if we peel it off, it won't be to shelve it. And I don't know that I would sell all of the cancer, but maybe breast cancer, lung cancer, some others, and still leave some of the brain and stuff for us to do. But the answer is yes, but with the caveat.
Gotcha.
And my last question is, after this round, how much more money will you need to raise in order to get to the FDA approval?
For the IND or for the to go to marketing?
To go to market.
So, I battled this with people for a while, with investors. When I was at AbbVie, if you do orphan diseases, you can do a phase 2B. And that's our goal, is to do orphan diseases where we have metastatic breast cancer or late term COVID, where we could get an EUA even for that, especially in Europe, that hasn't not declared there's no emergency. So we would like to get a 2B and to go into the market at that point.
Great.
So I'm sorry, to answer your question, that would be around $10 million. Sorry about that.
10 million then. Okay, great. It's good to hear. Well, that takes us to the end of our time here. I want to thank Anita and Donald for your great information there. Let's go ahead and bring our next presenter up. Let's have James Blackledge of Capella Imaging come up and show your slides, James, and kick off when you're ready.
Okay, thanks. I'm Jim Blackledge. I'm the CEO and one of the co-founders of Capella Imaging. We're a clinical stage company located in St. Louis, Missouri, that grew out of the Washington University Medical School. The founders have a great deal of experience in drug development, from discovery to commercialization, and also one of our founders is a cardiologist at the Washington University Medical School. And I want to talk to you today about our lead candidate, which is a fibrin-targeted nuclear imaging agent for looking at thrombus associated with LVADs and intracardiac thrombus. Oh, goodness. So there are two things driving this problem. The first one is clotting that is associated with the left ventricular assist devices. So heart disease is responsible for about 20% of deaths in the US. Many of these patients that are on the list for a heart transplant are fitted with LVADs to provide them with mechanical support, either until a donor heart is available, or as I'll show on the next slide, actually just to sustain them for, until the rest of their life. Technology has gotten significantly better over the years, but all these devices are still associated with thrombus, whether it's actually in the pump itself or it's in the quiescent dead regions of the surrounding heart that it's trying to support. And this results in one of the major side effects of these are an elevated risk of stroke. To counter this risk, patients are treated with heavy levels of anticoagulants, and that is its own problem. It induces gastrointestinal bleeding, and about a third of these patients are going to have extended hospital stays to treat this bleeding. 50% of those patients are going to have repeated hospitalizations. The challenge is presently there is no way to assess the clot load within these pumps. They're titanium shells, and there's no biomarker to assess the effectiveness of the anticoagulation treatment. So essentially, patients are dosed until you walk them up until they start bleeding out of their eyes, and then you back it off a bit, and there's no way to assess how well it's doing. The other problem is that the rules for allocating hearts have been changing. In 2018, they were changed. They're about to be changed again. Typically, there are about 5,000 patients on the heart transplant list. However, it's getting difficult to get on the heart transplant list. There are many, many more patients that actually need a donor heart. Initially, LVADs were used for so-called bridge to transplant. These days, the primary use is so-called destination therapy, meaning you're going to live with this device. And the growth rate for LVAD is projected to be an 18% annual growth rate for the next several years. However, one of the challenges is this post-surgical support and the cost of quality of life and how to deal with the bleeding issues and the potential thrombus. And this is where we think Fibricent can step in and play a significant role in addressing what is right now an unmet need. So this is sort of a cartoon image of our compound. It's a tetramer. Each of these arms is made of repeating polyethylene glycol units terminated with 2 peptides. One peptide binds fibrin exquisitely tightly. The other peptide chelates the radioactive technetium. We've already done all of our pre-IND enabling activities, and some of those are summarized here with the protein binding, the clearance, et cetera. What you see on the far right is an image of an LVAD that has been removed from a patient that came in for pump exchange. And it has been implanted in a 70-kilogram calf, which is what we use as model for humans. So this is an actual human pump with human thrombus that has been imaged with Fibricent. And I really want to stress this. This is as close as you can get to clinical data in a preclinical model. So let's talk about the market opportunity. There's about 600,000 patients in the US with advanced heart failure. And as I mentioned before, the numbers are difficult to tie down in terms of how many people on that in that list end up on the transplant list. And the rules are constantly changing. But if we assume a rather conservative estimate that maybe 1% of these patients are eligible for LVAD emplacement, and again, a very conservative estimate of $2,000 per dose. And I'm not going to walk through all of the numbers here, but in our vision, this is sort of a maintenance. Patients would be imaged twice a year to assess if they're developing thrombus within their devices. Patients that are having gastrointestinal bleeding and need to have their anticoagulant doses would be treated more regularly. But we're estimating about $36 million annual revenue, and that growing to about 70 in 5 years. Now, these numbers, in terms of patients, place us in the orphan drug area, which is something we are looking to exploit. And I won't go into that here, but most of you are probably familiar. There's a lot of benefits to that, and it enables us to pivot into other markets that we're very interested in, such as pulmonary embolism, acute coronary syndrome, prosthetic valves, other things that have solutions or solutions that we think we could improve on. So competitors in this space, CT and MRI are just not possible with this indication because of the metal pump. There is a Boston-based startup, or at least it's something that came out of the university there, that is working on a similar product that is based on PET imaging. We are using SPECT, and for things that I won't go into here, we think that SPECT is much more universally applicable and is more amenable to general implementation. The follow-on indications, our competition is really standard of care, and most of these are focused on eliminating potential. If you present as chest pain, you'll be treated with some of these imaging modalities to try to eliminate other possibilities, and what's left is often called a pulmonary embolism. Key concepts I want to stress here again is our preclinical model is as close as one can get to clinical data. And also this is a nuclear imaging agent. So the amount of material that is dosed is exquisitely small. We've already done a GLP toxicology study. We have a clean bill of health from that, but these are the two areas where drugs typically fail going from preclinical to clinical: efficacy or tox. And this drug, Fibricent, is highly de-risked in both of these regards. This is the Capella imaging team. Myself, Dr. Greg Lanza, who is the cardiologist from Washington University Medical School. My partners, Bill McGee and Grace Coy. And again, we have a lot of depth and experience in drug development through the whole, from discovery to commercial. These are the guys I really want to brag about. These are our medical advisors. Barry Siegel, I always date myself with this reference, but he's like when E.F. Hutton talks, people listen. Barry is just a household name with the FDA and nuclear imaging. Same as Sally Schwartz. She has written a number of the guidance documents. Greg Ewald is in charge of the heart transplant program. All these people are heavily invested and supportive of this program. We have exclusive license of the intellectual property for this through the Washington University Medical School. Additionally, we have made some improvements during the development of our scalable GMP process during our pre-IND activities that will extend that patent. And we are presently looking for a $5 million round, which will drive us through our phase 1 clinical trials. This is based on a $10 million valuation that was done by BDO when we closed our Series A. North Star Medical Radioisotopes invested $1 million in the company, and that is what has driven our IND enabling activities. They are actually expressing interest in potentially acquiring the asset once it's ready. But I think that's the last slide I was going to show you. So, yeah, thank you. I'm happy to answer your questions.
Great, James. Thanks for that. Eric, what are your questions?
Yeah, I guess, appreciate the presentation on this one. Curious from an investor standpoint, I know you guys came out of Washington University and you had a slide that showed that they owned the IP. Kind of wonder if you could talk about how that works, what royalties or any you have to pay to them, kind of what the structure is like.
Yeah, so there is a big document, and they do get royalties and a percentage of any annual sales. And as you can imagine, it is a dense document. So they do have a large piece of the pie, but we have the exclusive license to work with whoever we want to market this to.
Okay, thanks.
And Laura, what questions do you have?
Yeah, given that it's a smaller market than some of the other companies we heard today, I'm curious like what the clinical trial recruitment process looks like for you, and if you're planning to like leverage AI in any aspect of that.
Well, yeah, AI is interesting. We're not really implementing that, though that there may be interest in that in the future. Because it is a nuclear imaging agent, the clinical pathway is somewhat different. Clinical trials, the numbers for us, we're looking at about 28 patients for a phase 1 clinical trial, 56 for phase 2. And the other interesting thing is, the response, and the reason you can get away with this is the response is essentially binary. It's an image. It's not that you're having to move the curve and you've got to power it with huge numbers to demonstrate that you actually have efficacy. So we have partnered with a superb company here in St. Louis that is got tremendously deep in nuclear imaging clinical trial work, and we're currently setting up a pre-IND meeting and moving forward with this.
Great, thanks. My question, James, is does this procedure require the physician to change their process or behavior in any way, or does it just directly fits in what they're doing?
Well, so what would happen, the way we envision it is, at least initially, is, I guess to answer your question is yes, it would require a change in behavior because right now there is no diagnostic tool. The way we envision this is a patient comes in, let's say they're a new LVAD recipient. The device would be implanted, they would be in the hospital, they would be on anticoagulant treatment, they would be screened with fibricin, and you would establish a baseline level showing there is no thrombus accumulated in the device or in the surrounding heart. I should say that the newer generations of LVADs are significantly better than the previous generation. So the bigger risk is really the accumulation of thrombus in the dead regions of the heart around the LVAD. And what happens is when those come through the pump, they're ejected, and that's what leads to a stroke. So the change in behavior would just be using this tool. You would schedule, and to some extent, we still have to work out the best way to do it, but we envision at least every 6 months that a patient would come in and be imaged and assessed for, how well is this level of anticoagulation doing. If there's no sign of clot development, but they're still wrestling with gastrointestinal bleeding, you have the potential to titrate that treatment level down.
Great. I guess my question is before, what's the remaining amount of funding you need to get to FDA approval from after this round?
So the key opinion leaders involved here feel that it's very likely that, again, because the nuclear imaging aspect of this, that we will not be required to do a phase 3 study. And so what we anticipate is there will be another $5 million raise towards the end of next year, which will drive us through our phase 2 trial. And at that point, we would look to exit.
So you would exit before you go to market then?
Yeah, we're chemists, and we've been in this. This is not our first company as a group of collaborators here at Capella, and so our passion is really around the development and getting it to the patients. We're less passionate about being businessmen, and so our goal is to show that this has value, show that it can impact patient health, and take it to somebody else that has the infrastructure, the sales team, the quality, the regulatory affairs parts that are necessary to actually get a product to market.
Okay, that sounds great. Thanks, James, for doing that. Let's go ahead and close this down, and let's go to the next one. Jeff Missling of Spentera. Jeff, if you can go ahead and put your slides up, we'll kick off with you next.
All right. Good afternoon, everyone. I'm Jeff Missling, founder and CEO here at Spentera. So at Spentera, we are creating injectable drugs utilizing our differentiated and proprietary sustained release technology that we describe as a microsuspension. We have directed this technology to our lead asset, SX600, and have best-in-class phase 2 clinical trial data for lumbar radicular pain, or sciatica, which is a $3.6 billion market opportunity in the United States. Our product is highly differentiated from the competition and well-positioned commercially in this field. And again, this is enabled by our microsuspension platform technology that can deliver an ultra-high concentration injection through small needles and catheters of a sustained release formulation. And this provides multiple high-value follow-on applications. One thing that's unique about Spentera, we have a highly capable formulation and manufacturing team. In addition to clinical and regulatory, we have our own cGMP facility. We have developed and now manufacture this product in-house, and our facility can take the drug through phase 3 clinical trials. So why do I call our drug best in class? Here's a high-level summary of the data available in the field. Our drug, in a recently completed clinical study, had a 71% treatment response rate at a 60-day primary endpoint. And I'm going to show you the data that was actually maintained through the full 180-day follow-up. Generic drugs that doctors have largely abandoned had the best reported outcomes of a 54% treatment response rate. And our main competitor, a company called Silex, which is currently sitting out there with a market cap of about $800 million after completing a single phase 3 clinical trial, had a 38% treatment response rate at a 28-day follow-up in their phase 3 clinical trial. So our data stands apart in the field. Back pain, let's talk about it in general. It's the leading cause of disability around the world. Everywhere on this map that you see a dark purple country, back pain is the leading cause of disability. It's also the leading cause of disability for actively deployed military personnel. And the DoD has actually provided us with $7 million of grant funding to support the development of our drug. It's also the most expensive condition to treat in direct healthcare dollars. $135 billion a year, more than diabetes, more than all forms of cancer combined. And if you look at the treatment of neck and back pain, only 3.5% of the spending is in pharmaceuticals. All the spending is going to surgical and spinal cord stimulators. So let's talk quickly about the patient journey here in back pain, right? If somebody has a new onset of back pain, they're going to try oral medications, physical therapy, or simply waiting it out. If their pain persists, they'll often have an MRI. If that MRI shows a herniated disc in their spine, they're a prime candidate for an epidural steroid injection. This is simply a cortisone shot targeting a nerve root adjacent to a herniated disc in your spine. It's one of the most common medical procedures we perform, 10 million annual procedures in the United States alone. It's done in every major country around the world. And these are always attempted before a patient would move on to expensive surgeries or spinal cord stimulators. But what's wrong with the drugs doctors have today? There are all generic steroids that are being used in this setting. These drugs are off-label. They're not FDA approved for this purpose, and they're not approved in any country for this purpose. Moreover, in the United States, they have an FDA warning statement on them regarding some safety concerns of that off-label use in the spine. And more importantly to patients, the outcomes are not very good. Of those 10 million injections we perform, 40% are repeat injections, a second, third, or even fourth injection in the first 90 days of treatment. So we're having low efficacy rates, short durations of action, and repeat procedures are extremely common. So what we've done here at SpineThera is actually quite simple. We've taken the corticosteroid that doctors prefer to use today for safety, dexamethasone, and we have formulated it into our microsuspension platform. These are biodegradable or bioresorbable microspheres that are half the size of a red blood cell, so only 3 microns in diameter, and they are non-aggregating. They will not clump together. They're made from a very well-known excipient, PLGA. So these little microspheres enable sustained localized delivery in a safe manner right where they're needed at that nerve root in the spine. Here's the data that demonstrates the difference from the generic drug that's being used off-label today. You can see here in the blue curve, if you give an epidural injection of the available dexamethasone, the patient will only have about 2 days of drug exposure. If you give a single epidural injection of our sustained release formulation, you'll have about 90 days of drug exposure from that single injection. And again, this is happening locally at the site of injection, just adjacent to that nerve root. So I want to share with you how this translated into improved clinical outcomes. We recently completed a phase 2 clinical trial in 56 patients. Patients were randomized into either a saline placebo arm, a 12.5 milligram low dose of our sustained release formulation, or a 25 milligram high dose. We followed these patients for a full 180 days. Here is the primary outcome data. We were measuring the proportion of patients that had a 50% or greater improvement in their pain score from baseline. Patients were reporting a pain score every day in a custom smartphone app. So we had real-time pain reporting. You can see here at the pre-specified 60-day primary endpoint, 71% of our high-dose patients hit that treatment goal, whereas only 13% of the placebo patients did. This is highly statistically significant in this small phase 2 study. You also notice that the high-dose patients maintained that efficacy at 64% or higher through the 180-day follow-up. So again, 2/3 of the patients here had significant pain relief from a single epidural injection over 180 days. We have a strong intellectual property portfolio with 5 issued US patents, patents issued in all the major pharmaceutical markets around the world. And I also need to point out that there is no pathway in the FDA to get a generic version of a locally acting sustained release drug approved. The only competition we will face are from other branded innovative products. Let's look at the market here. As I mentioned, we do 10 million epidural steroid injections a year in the United States. Our clinical development is targeted at the largest segment, lumbar, sacral, transforaminal epidural injections. That's kind of a word salad, but again, we're going after the largest segment of the market, and that's what our drug labeling will be directed to. 58% of the market is there. So of those 5.8 million procedures, if you move to the upper right here, we will eliminate 40% of them, right, with the outcomes of our sustained release formulation, leaving 3.5 million procedures available. The KOLs in this space who are following us are telling me they expect our drug to become standard of care when it's available, because the current drugs are off-label and warned against. We estimate at a 50% market share, $1 billion of peak sales for our drug at a $600 per dose price, which is well supported by analogous drugs in the pharmaceutical industry. There are 4 known competitors in this space, other companies chasing the same large market opportunity. I will just say we are the only company with a 90-day sustained release drug using an active ingredient with a proven mechanism of action. The others are all either short-acting drugs with no improvement over the generics, or they're exploring a new compound that has no proven efficacy in this setting. Today, I'm here raising a $5 million bridge round. The use of the proceeds here is directed at a FDA-required mini pig epidural toxicology study. We've already completed toxicology studies in rats and canines. The FDA learned on our dime that rats were not an appropriate model for this route of administration, even though they agreed to that at our pre-IND meeting. And they've asked us to go back and do this mini pig epidural tox study. That's what we're raising money for today. It's essentially a check the box exercise, repeat what we've already done in dogs and the FDA has accepted in miniature pigs. The convertible note is 10% interest. It's going to convert at the lower of a 20% conversion discount or $1.08 per share, which is a $24 million valuation cap. It comes with a 4-year warrant, and we're taking minimum of investments of $50,000. On our team, I will highlight our Chief Medical Officer, Lee Techner, with 22 years of drug development, largely focused in analgesics and pain therapeutics. On our board of directors, we've got 2 notable pharmaceutical executives, Doug Drysdale and John Foster. Our KOLs on our medical advisory board are on the top of the field, including David Mauck, the chair of pain medicine at the Mayo Clinic in Rochester, Minnesota. Let's talk about exit scenarios, right? We've completed a phase 2 study. We're essentially 1 non-clinical study and 1 phase 3 clinical trial away from FDA approval. This positions us well for an IPO when the market rebounds, or partnering with a large established pharmaceutical company either during or after our phase 3 clinical trial. Valuation analyses and comps would show an 8x return potential in 2 years and a 24x return potential in 4 years. With that, I'd be happy to take your questions.
Thank you, Jeff. Laura, what's your first question?
Yeah, I mean, it's pretty ambitious, I would say, return multiples. And given the competitive side, some are just more advanced in terms of commercialization. So I'm curious, like, how do you plan to capture their share of the market?
Well, it's interesting, Laura, because on the competitive slide, Solus Therapeutics has already failed a phase 3 clinical study. They spent $50 million of Deerfield's money on that, and they've been recapitalized to make another run at it. So they're no farther ahead of us. The other competitor, Silex, our competitive intelligence is telling us the FDA is not satisfied with their phase 3 study, and we think they're also going to have to do a do-over. So we actually believe we're all at the same point, which is 1 phase 3 clinical trial away. Part of the reason the return potential here is high is I think we're offering an extremely attractive $24 million valuation cap in this convertible note round, which is just reflective of the current market conditions.
Great. And Eric, what questions do you have?
Yeah, I guess kind of following up on that a little bit, is it, so if I understand it, so is this a bridge round to the IPO? Is that what you're saying? You need 5 million now, and then the next would be the $60 million offering, or is there going to be more capital raises after that?
There would be more capital raises after this, Eric. So we would need to raise significant additional capital or partner the drug to move it into the phase 3 clinical trial. But we know we need to conduct this mini pig tox study to satisfy the FDA. And we feel like it's a nice opportunity to bring in some high net worth family office type money, maybe small boutique VCs to check that one box off the list. As we're out, I know it's going to just take us a little bit more time to raise the capital needed to move into that phase 2/3 pivotal clinical trial.
Great. My question is, it came up during the IP slide. A question from the audience came in about IP protection in China. Do you have that? Do you think that would actually hold up?
So we are not actually issued. I'm sorry, I'm flipping through my slides trying to get back to that one. We are not issued in China right now. We're in progress in China. So I'm optimistic we'll get our claims allowed there, but that is the last country we are working on, actually.
Okay. And my other question was about physician change in process. When they use your therapy, is it going to change the way they handle patients in any way?
It changes nothing about their procedure at all. They simply will use a vial of our drug instead of the generic drug they're using off-label today. And this was the case in our phase 2 clinical trial that was performed in Australia. It was just seamlessly integrated into their standard workflow. It changes nothing about the procedure.
Okay, and where does the IP originally come from?
It's all in-house. I'm the lead inventor on most of these patents, and it was completely developed in-house.
And what was your background before you did this?
So my background is in chemical engineering, actually, and I've spent about 20 years in the pharmaceutical industry focused on sustained release formulation development. I previously worked at a company that had a corticosteroid implant to treat diseases of the back of the eye that we licensed to Merck. And I've basically spent my entire career making what I would describe as functional polymeric materials. My first job was making solid state polymeric batteries at 3M, if you can believe that. It has a lot of analogies to sustained release drug development. So my background really just positioned me to make this concept reality.
Great, thank you. Appreciate
Thank you.
that, Jeff. If there are any more questions, we'll go ahead and move to the next presenter. We'll have Ping Yi of Voxy come up, and he'll be the final presenter today. Ping, if you go ahead and share your slides.
All right. Hi, everybody. My name is Ping Ye, CEO of Oxy Health. My background is in engineering, nanotechnology. Spent about first 15 years of my career in the tech space. And after surviving blood cancer, switched over the last decade in the world of life sciences. Voxy Health is a company I joined over a year ago. It's a spinout from Boston Scientific. The name comes from extracting insights and intelligence from VOCs, which are volatile organic compounds from the human breath. So yes, this is a spinout from Boston Scientific. It's where I am right now, as well as University of Minnesota. Both organizations share in 14% ownership in the company. Our vision is to save millions of lives through early detection using breath diagnostics. That is the technology that the team has developed the last near decade. Our mission is to create the most accurate, approachable, and accessible diagnostic in the world. There are 26 granted patents on this technology. $11 million has been put in to get to this point in time. This has been tested on a couple hundred people in 3 clinical studies and shown great accuracy. So we're excited to bring this to commercialization. We're currently raising a $5 million safe with 20% discount, 20 million post-money cap. We're actually, as of yesterday, closer to 80% filled in this round. We have a syndicate as shown here, and the use of proceeds are to commercialize this patented breath diagnostic with a strategic partner that I'll talk about in a few minutes. I think everybody's been touched by cancer in some way. I'm a cancer survivor, as mentioned before. In the space of lung cancer, 84% of lung cancers are diagnosed too late. Still the #1 cause of death, still a low screening compliance. 95% do not comply with free annual screenings. 10 times improvement in terms of survival if found early, and still a huge strain on the US economy, in addition to the impact it has on families. So in terms of the questions that a device like ours can answer, from a screening perspective, is someone cancer-free? Number 2, are nodules cancerous? Oftentimes, about a third of the time when biopsies are done, it's non-cancerous, and being able to understand that beforehand would be huge for certain organizations. And then the last 3 questions are around monitoring and surveillance. Our device will first be focused on risk stratification, in that identifying if nodules are cancerous or not. One of the things that really intrigued me about the team here at Boston and what they had developed was they didn't specifically design a technology to look at one particular disease like lung cancer. It was actually a platform to look at the full spectrum of volatile organic compounds using one device. So this one device can be used for a whole multitude of diseases, with the unique algorithms being the differentiator. So the hardware basically stays the same on everything. This is, we did not invent or discover VOCs. This has been leveraged for decades of work, has been leveraged here at the company. The diagram on the right shows how many different disease states can be studied using VOCs. And the team's basically developed a cross-reactive array, or an array of sensors that can look at multiple diseases and the metabolic response of those perturbations on the body. And so whether it's cancer, age-related diseases, or infectious diseases, those all have been proven in hundreds of peer-reviewed journals that they exist, and there are shifts in VOC concentrations when that happens. So over the last near decade, the team's prove the ground truth using time of flight mass spectrometers, taking chemicals coming off of tumor cells and flowing them and proving that this does have part per billion sensitivity. We have a clinical desktop form factor currently that's been through the clinical studies. Here we're highlighting the actual workhorse, which is this varactor or variable capacitor. And we're now at the commercial design, which is basically a breathalyzer. The printed circuit board's about the size of a stick of gum. And there you can see the relative size with respect to a quarter. And the workhorse essentially is the ability, and a lot of the innovation is that they have figured out how to put a single layer of carbon on top of a nanocircuit. And on top of that, they figured out how to scale and commercially build on top of that single layers or self-assembled monolayers of receptor chemistry that can pick up all the families necessary to get the full spectrum of volatile organic compounds, which the team uses to create algorithms to bifurcate populations and then identify certain subgroups within disease populations. Here's an example. This is work done by another group, but it shows that there's different concentrations of chemicals from different cell types of lung cancer. There are multiple families. What the card does is it can measure different families of chemicals. So if it's 2-butanol, it picks up those 5 chips of 2-butanol, respond to 2-butanol in unique ways because there are over a dozen parameters within the quantum capacitance response that shifts. And so from that, they can measure all the different chemical families and produce these very rich data patterns, which we use machine learning to reverse engineer based off of whatever disease group we're looking at, in this case, lung cancer. So specifically lung cancer, here's a great example of how volatile organic compounds shift. And this really opened my eyes when they first showed it to me. So here, 33 compounds statistically shift in abundance percentage or concentration, and 9 new VOCs are produced when one has lung cancer. And this device is able to measure all of them with 1 minute of breathing. There are 26 granted patents and over 30 filed around this technology. Most of them is around the varactor or variable capacitor that picks up the quantum capacitance, and then also the many different functionalizations that picks up the various families that relate to the disease VOCs. So this actually works. We've been doing this for several years now. The team has at least 3 clinical studies, 200 people, with regards to sensitivity and specificity in the 90s. This is significantly higher than the handful of other breath companies out there in the world. And uniquely, the stage 1 accuracy is many times better than liquid biopsy, the cell-free circulating tumor DNA approaches, because this is a more of a metabolomic approach. And we expect these numbers to increase and improve over time. So one way to view the uniqueness of this approach is through a radar plot. So I'm just showing 3 parameters here of those capacitance shifts. But what you're looking at here are those blue circles, which are the healthy controls versus the lung cancer disease in the orange plots. And we can very clearly, with high confidence, with a p-value of 0.0001, predict the differences between healthy and disease. So from a competitive advantage standpoint, in summary, this is non-invasive, 1 minute of breathing. It can be performed anywhere. There is no lab, it's real time. There's no data loss. We do not ionize the samples because we work in full breath humidity, which everybody else does not. They have to remove the moisture, which removes a lot of the data. And there's no radiation involved, such as with CT scans. Here's a more detailed view of the competitive landscape. Essentially, everybody else is a mass spectrometer that's been around for a while. This is basically a mass spectrometer in your hand, but with the added benefits of being able to work in a full human environment and really a form factor and a cost that is significantly better. So 10 times better in size, over 20 times better in cost per procedure. It's just a big leap forward. With regards to aligned interests, of course, you can imagine this would bring peace of mind for patients who don't have to wait and see, which is often the times when you have tumors smaller than 8 millimeters. We can inform patients and their providers, as well as lower the cost for payers. So our path to commercialization quickly is from step one is to leverage our desktop unit to work with a health system in Latin America. They have actually 650,000 high-risk lung cancer patients that they screen annually. So we'd like to get involved with their patient group. And then once we build confidence with our algorithms after testing a much larger sample size of individuals, we'll then move to FDA. We can imagine doing this in the second half of 24 with approval in 25. We do expect a Series A early next year around May. And then, of course, based off of the data, we'll approach the reimbursement codes based off of how the data looks. The team's done dozens of approvals. I'm super lucky to work with a team as experienced as this, over 100 years of life science experience. I'll just highlight a couple individuals. Greg Sherwood, the original kind of scientific founder of Boston Scientific, senior fellow, has left to join me at Voxy Health. Same with Rhea Finns, our chief of product. Randy Schistel is actually the global head of R&D for all of Boston Scientific, and he just joined full-time 3 weeks ago. So it's really a phenomenal team that can bring this forward. We're raising $5 million, as mentioned before. Major milestones are to bring the commercial desktop with our strategic partner and partners and launch this LDT effort to drive more data, really lock down the algorithm, drive early revenue to set us up for FDA approval in 25. As you can imagine, there are a lot of potential acquirers in this space, from health providers, consumer health companies, tech companies, retail and medical device, and already some of them are having conversations with us. So thank you. I think that's the last slide.
Great. Thanks, Ping. Eric, what's your first question?
Yeah, I'm curious, do you have to baseline this as you use it for different patients? And then how does that work? Is it a one-time use, or do patients then use it over a period of weeks or months before they get a diagnostic or enough data?
Yeah, so there's two questions. So the first one is, it can be one-time use and then recycled, or because this is not a chemical reaction on the sensors, it works at a quantum capacitance level. It actually can be reset. So in our current desktop units, we reset them dozens of times. So even though the cost might be $300, you can divide that by 30 and get a pretty low procedural cost. With respect to baselining, we have two approaches. One is we can compare the individual to the subpopulation that they're being related to, right, to see where they are relative to those individuals. But as you can imagine, from a personalized medicine standpoint, one could get the first baseline measurement, and then 6 months or a year later or 3 months later, whatever the time frame is, they can relate that next breath print back to themselves on a different device. That's how we imagine it working.
Great. And Laura, what's your first question?
Yeah, in the same vein, I mean, you mentioned the accuracy is significantly higher. So I'm curious the other accuracy levels that you're benchmarking against.
So I think I had on that one slide, but essentially they're in the 70s and low 80s. I think it was on this slide right there. Oop. Yeah. So Owlstone, for example, is in the 70s, Pulmostic 70s, Enos 80s. They may have a new paper out with higher sensitivity, but I think their specificity was like 50. So one of the differences, because we're not looking at 3, 4, or 5, or even 10 biomarkers, we're looking at 2,000 VOCs in terms of the full spectrum of it. So that's what really gives us kind of a higher accuracy.
Great, thanks. And I have 3 questions from the audience. The first one is, how unambiguously can you decode a cancer versus no cancer breath sample? How many samples do you take?
Yeah, so it takes about 100 to 150 in kind of samples. Right now, even with fewer than that, the signal is so different that we're still able to have the p-value confidence that I showed before and being able to bifurcate populations. We actually expect that the signals will be so unique that it's not just cancer, no cancer that we're looking at, but differences within cancer, and then within cancer, like lung cancer, being able to tell between squamous cell and adenocarcinoma and various cell types. And then we're already starting to see likely some differences between stage 1 and stage 4 late. So the more we test, we'll see and have more resolution.
The next question is, how do food and beverage in the breath pollute the results?
It actually doesn't. We get that question a lot. They spent the last decade figuring out what are the VOCs, particularly ones smaller than 160 Daltons in size, that are related to disease. And so that's what our scientific advisor, actually the original inventors of the sensors, came from the University of Minnesota. And one professor, Dr. Bowman in particular, specializes in molecular sensing. And so he spent his last decades doing that. So together, the Boston Sci team worked with him and another professor to identify which, to really tune which chemical receptors are needed to get the right VOC spectrum. So actually, after hundreds of self-testing here, it hasn't been affected by coffee or chewing gum or anything like that.
And the last question is, what is the
I wouldn't
next?
smoke when you do it though.
Okay,
I would take
that would change it. Yeah. What is the next follow-on therapy after they take this test?
The next follow-on therapy or the next follow-on disease after lung cancer?
I believe it's the therapy is what treatment do they go through once they have this? Is it related to yours or is that separate?
Yeah, so in turn, I'll say therapy or treatment solution. Maybe I think a couple things. The first is that in our first intended use, which is to understand if a nodule is cancerous or not. If it is, but small and not metastatic, you could do a simple surgery. So it might not actually be an expensive therapeutic. And that's actually kind of the point. When you consider that immuno-oncology drugs are over $100,000 per year, and let's say the person's on it for 3 years, that's, over $300,000 in cost to the payer versus a one-time $15,000 cost of surgery. That is a massive difference in cost, claims costs to the payer, which really aligns everybody in an approach like this. And so that's why we're pretty excited about this, because this has a broad influence on so many different stakeholders in the health system. Yeah.
So would you consider these tests suggestive or conclusive?
Suggestive or conclusive? Well, what we do provide is a probability of cancer, and that's the readout that will provide the physician. And so there's always a level of probability to any prediction. One ways we can influence it is through confidence learning curves and in our machine learning approach, and to be able to actually choose a higher confidence to further drive up. Actually, the numbers here haven't even been driven up with that approach. So if we applied that approach, we can likely get even higher on our accuracy. If we do, the only drawback is you have a larger undetermined group because you've chosen to have high confidence in the prediction.
Okay, great. That answers our questions for today and concludes our presentations. Thanks, Ping, for your presentation.
Thank you.
I want to go ahead and thank all the presenters today, everyone that pitched today. Appreciate your taking time to prepare. It was great presentations. Everybody stayed within time as well. That's a record. And want to thank Eric Alfuth and Laura Hamilton for joining today. Appreciate that, guys. And so we'll be passing out the results as soon as we have them, and want to thank everybody for the time. With that, we'll go ahead and wrap up for today, and we'll see you guys next time. Have a good one.